Okay, so imagine for a second all the wiring inside your brain. If you were to stretch out all those nerve cell connections, the axons, from end to end, you'd have something like 300,000 kilometers of cable.
It's an incredible number.
I mean, that's enough to wrap around the entire Earth 4 times.
And that right there, that is one of the fundamental problems in all of neuroscience.
Yeah.
You have this massive sprawling network. So how does the brain make sure that a signal traveling a really short distance and another one traveling a comparatively enormous distance?
Right, from one side of the brain to the other, or even down the spinal cord.
Exactly.
Yeah.
How do they both arrive at their target at the exact same millisecond?
It's the ultimate timing problem, because if those signals are even a tiny bit out of sync, our whole perception of reality, what we feel as a single coherent now, it just shatters.
Precisely.
Hmm.
If you look at it from a physics perspective, Richard Feynman had this idea that time isn't some abstract thing that flows. He defined it as the ordering of events. And that's what the brain has to do. It has to create and maintain that strict temporal order across these huge neural distances. If that order breaks down, if the time cost of space gets too high, the brain just can't perceive a unified moment.
All right, so let's really unpack this, because the main mechanism that manages this feat, that minimizes that time cost, is this one critical fatty structure, myelin. And for this deep dive, we're focusing on some really fascinating new research, mostly from the Cole group, that details the hardware of this biological clock, and crucially, what happens when it breaks down.
We're basically looking at the architecture that holds your moment-to-moment reality together.
So before a signal can even travel fast, it has to start reliably, right? You can't synchronize a messy signal. So where does it all begin? Where does the action potential, the AP, actually kick off?
It all initiates at one very specific spot, a little non-myelinated junction near the cell body. It's called the axon initial segment, or the AIS. You can think of the AIS as the neuron's true decision center. It takes in all the inputs and decides yes or no whether to fire.
And what's making it so reliable? What's the mechanism there?
It comes down to this incredible organizing protein called ankyrin G, or just anchor.
Yeah.
Anky
Yeah.
is, well, it's like a construction foreman. It's an anchor that physically grabs and clusters all the critical sodium channels, the NAV channels, and holds them right there in the AIS membrane.
And why is that clustering so important right at that specific spot?
Because by packing them all together, ANKG makes sure that when the impulse is generated, it happens reliably, with very little energy, and it gives a precise, standardized starting kick every single time. It basically sets the clock.
Okay, that makes perfect sense. You need a perfect start for a perfectly timed race. But here's where the new research gets really interesting. We all learned that NAV channels are for, well, sodium, but the source material shows they let something else sneak through.
Yes, that was a genuine surprise in this study. They found an influx of calcium, K2 plus JAR. I mean, we know these channels are designed to be incredibly selective for sodium, but right at the peak of the action potential, these NAV channels, they also allow a tiny, rapid influx of calcium.
And that's happening at the AIS and also at the nodes of Ranvier, the gaps in the myelin.
Exactly.
How small are we talking here? I mean, what kind of precision did they measure?
The measurement showed the channel has only about 0.38% conductivity for calcium. It's tiny.
So it's overwhelmingly sodium.
Overwhelmingly. But that tiny fraction of calcium current is incredibly fast. It creates these sub-millisecond calcium signals right at those critical points.
Well, why does that matter? If sodium is doing all the heavy lifting for the action potential, what's this little flash of calcium doing?
It's all about regulation and timing. This was especially clear in these specialized cells, the layer 5 pyramidal neurons.
And those are the important ones, right? The
Yeah.
long-range communicators.
They are the primary long-range output neurons of the whole cerebral cortex. They're sending signals across that 300,000 kilometer network we talked about. So this little calcium flash, it seems to act as a local timer. The thinking is that this precise, say, K2 plus entry might regulate how the NAV channels themselves work, or maybe it opens up other key channels like BKCA channels.
Okay, hang on. Let's just quickly define that for everyone. BKCA channels.
Right, so BKCA stands for Big Conductance Calcium-Activated Potassium Channels.
Ah, calcium activated? There it is.
There it is. So the idea is that this tiny, super-fast calcium flash immediately opens these potassium channels, which then rapidly resets the membrane. It's a whole cycle that confirms the start of the electrical code is controlled down to the sub-millisecond level. It makes sure the neuron is ready for the next signal almost instantly. It anchors the whole system.
Okay, so we've got a perfectly timed, reliable start to the signal. But that still leaves us with the huge problem of distance. Once that AP fires, it might have to travel a meter or more without losing time. And this is where myelin comes in.
Myelin is, it's basically a biological space-time compressor. It's truly amazing. In early nervous systems that didn't have it, the signal just sort of propagated continuously. It was slow.
Like electricity in a leaky hose.
That's a great way to put it.
Hmm.
Myelin is the insulation. It's a fatty sheath that wraps around the axon and stops that leakage.
And that changes everything about how the signal moves.
It really does. It
Yeah.
forces the propagation to become saltatory.
Meaning it jumps?
It jumps. The action potential effectively leaps from one tiny gap in the myelin, a notoranvir, to the next.
And that jumping is what gives you that massive speed increase, the 10 to 100-fold acceleration.
Absolutely. It's like upgrading from a country lane to a fiber optic cable.
Yeah.
You need that speed for long-range synchronization.
Yeah.
There's no way you could track complex simultaneous events without it.
So the technical term for this goal is isochronicity, right? Myelin exists to ensure isochronicity, to make sure signals traveling different lengths arrive at the same time. It cancels out the time cost of distance.
And that simultaneous arrival is what allows the brain to create that coherent now. The whole way we process our senses is built on a mechanism neuroscientists call coincidence detection.
Okay, give us a concrete example of that. Coincidence detection in action.
So let's take the circuit the researchers were studying. A mouse's whisker touches an object. That sensory input zips to a relay center in the brain, the thalamus. But for the brain to correctly process that touch as a single meaningful event, it needs cortical feedback coming from those long-range layer 5 neurons we just talked about to arrive at the exact same time.
And when you say same time, what kind of window are we talking about?
A very, very tight window. The source material shows it has to happen within about 5 to 30 milliseconds.
30 milliseconds, that's it.
That's it. If the cortical feedback arrives outside that tiny window, the two events, the touch and the interpretation, are treated as separate things. The brain just can't put them together correctly.
So myelin is the physical hardware that makes sure the signal from the cortex makes that 30-millisecond appointment no matter how far it has to travel.
It's the conductor of the orchestra.
This all sounds so perfectly precise until it breaks. And the researchers modeled this breakdown with a treatment called cuprizone, which causes demyelination in a way that's very similar to lesions you see in multiple sclerosis.
And the results were just a dramatic shift towards what you could call neural entropy, a total loss of temporal order. Physically, the conduction velocity just plummeted. In those long-range axons, it dropped from about 1.1 meters per second.
That's pretty quick.
A respectable speed, yeah, down to about 0.35 meters per second.
Just think about that for a second. That's a 70% drop in speed on the network that defines your reality. If your Wi-Fi dropped that much, you'd be furious. But in the brain, the lack of speed isn't even the biggest problem.
Not at all. Speed loss is bad, but it's the inconsistency that truly wrecks the network. They saw two key timing problems. First was just increased delay. The signals were late. But the second, and much more damaging, was increased jitter.
Jitter, so variance, like static.
Exactly.
Yeah.
A huge variance in arrival times. It means the signal precision is totally gone. If your system depends on coincidence detection, a delayed signal is one thing, but a jittery signal is just unusable. The thalamus might still get the message, but it has no idea when it started or what the timing means. The order is lost.
The researchers had a great analogy for this. They said, missing a piece of myelin is like skipping the first black stripe on a barcode. The scanner, the thalamus, it sees the light, but it can't recognize the product anymore.
It's a perfect metaphor. The mouse can still feel its whisker being touched, but the brain can't exactly identify when or what.
And the cost of that is huge. This loss of coherence creates what's basically a low-pass filter on the brain's network. What exactly does that mean, a low-pass filter?
It means the brain can still handle slow, simple, low-frequency information, but it causes catastrophic failure when it tries to transmit complex, high-frequency bursts of action potential.
And those fast bursts are how the brain sends complex information.
Exactly. Simple tasks use slow signals. Complex thought relies on these fast, intricate bursts.
And when myelin is gone, those bursts just fail.
They fail spectacularly. The research actually quantified this. In a healthy axon, the failure rate for a single spike inside a fast burst was about 6%. With demyelination, that failure rate shot up to 32%, especially for frequencies over about 130 hertz.
So losing that high-frequency info is like, it's the difference between hearing a single, simple tone and failing to understand a whole symphony.
It is. The complex information is just filtered out. It's lost along the way. And this breakdown widens the brain's integration window. It takes longer to put inputs together.
Which leads directly to the cognitive issues you see in MS.
Yeah.
The disorientation, the trouble with high-level tasks, struggling to recall a name. The precision that creates our now is just gone, and the world feels fuzzy, delayed.
And there's one last layer of disorder here that we have to mention. The breakdown isn't just a passive slowing down, it actually changes the axon itself. They found that the demyelination process, surprisingly, could lead to an increase in that ANG protein.
The anchor protein we talked about at the start, it increases.
It increases.
Yeah.
It's like an unintended side effect of the damage. And this hyperclustering of the sodium channels completely upsets the neuron's balance. It can cause those damaged neurons to start firing spontaneously with no external input at all.
Wow. So if we put this all together, the message is incredibly clear. Myelin is the biological hardware that fights against neural entropy. It's what supports the brain's biological trick of creating a unified, immediate reality by canceling out the time delays caused by physical distance.
Absolutely. We have to remember the brain's sense of now isn't a given. It's a meticulously constructed illusion, and it depends entirely on these mechanisms achieving perfect temporal coherence.
And we saw how ANKA-G is critical for that precise start. But the research also showed that when myelin is lost, the signals don't just slow down. You get this secondary catastrophic effect of increased ANKA-G causing the spontaneous incorrect activity.
It's like the system starts generating its own internal static, its own noise, just cluttering up the communication lines.
And that raises a really powerful final question for you to think about. If myelin loss not only makes important signals arrive slowly and with high jitter, but also causes the brain to generate massive amounts of irrelevant noise internally through the spontaneous firing, how much of the cognitive confusion we see in these diseases is due not just to slow signals, but to the brain's own internal noise constantly interfering with the real meaningful information trying to get through? That's a powerful thought to take away. That was our deep dive into the myelin clock and the architecture of our coherent now. We hope you feel a little more well-informed.